welcome back friends. This is Dr. Varsha. Welcome to another video lecture from Botany Varsha. In this video tutorial, we will be talking about difference between eukaryotic and prokaryotic DNA replication. First thing you need to know what is DNA replication and why it requires inner life. The purpose of DNA replication is to produce two identical copies of DNA molecule. This is essential for cell division during growth or repair of damaged tissue. DNA replication ensures that each cell receives its own copy of the DNA. It conserves the entire genome for the next generation. First question is, what is DNA replication? The process by which the parent DNA molecule gives rise to two exactly similar sister DNA molecule is called DNA replication. First difference on the basis of the site of DNA replication. In prokaryotic cell, you can see this is bacterial cell in which DNA is scattered in the cytoplasm. So in prokaryotic cell where well-defined nucleus is not found and DNA is scattered in the cytoplasm. So the site of replication is cytoplasm in the case of prokaryotic cell, but in eukaryotic cell where well-defined nucleus is found and in the nucleus, nucleoplasm, genetic material, nucleolus, etc. proper things are found. So this is well-defined nucleus and DNA replication is found here in the nucleus. The site of DNA replication in the case of eukaryotic cell is nucleus. Second difference on the basis of the origin of replication. In prokaryotic cell, circular DNA is found and there is only one origin of replication, single origin of replication is found in prokaryotic cell which replicate towards both the side. Whereas in the case of eukaryotic cell in which long huge linear DNA is found and there are numerous origin of replications are found. In the picture towards the left side you can see the circular DNA. This is circular DNA of prokaryotic cell and with a single origin of replication. In the right side double-stranded DNA is found. These are parental DNA in which so many replication bubbles are produced. You can see many replication bubbles are produced in the case of a eukaryotic cell. In each replication bubble, two Y-shaped replication fork are produced. So in the case of eukaryotes, many replication points are produced. In prokaryotes, single origin of replication is found. This is the picture which is showing DNA replication in eukaryotic cell. Helicase enzyme, this is helicase enzyme which break the H2 bonds, hydrogen bonds which are found between the nitrogenous bases and a Y-shaped fork is produced in the case of eukaryotes, and this is topoisomerase which unwind the DNA molecule. Excuse me. And these are SSBP, single stranded binding proteins which are found in the form of tetramer, and because of that DNA molecules are stabilized. So in the case of prokaryotes, helicase break the bonds between the nitrogenous bases and two double helical structures get separate from each other, and DNA polymerase start the base pairing, and base pairing is complementary, and A pairs with T means adenine pairs with thymine and cytosine pairs with guanine only. And in prokaryotes, DNA polymerase third carry out both initiation and elongation during the replication process. And in the case of eukaryotes, initiation is carried out by DNA alpha, while elongation carried out by DNA polymerase delta and epsilon. DNA after DNA replication, DNA repairing and gap filling, two process takes place, and these processes are done by DNA polymerase 1 in the case of prokaryotes. But in the case of eukaryotes, same are performed by DNA polymerase beta. And DNA replication is very rapid in the case of prokaryotes. And DNA gyrase is needed for the process, and in eukaryotic cell DNA replication is slow. Sometimes it is very slow, and DNA gyrase is not needed. And DNA gyrase is an essential bacterial enzyme that catalyzes the ATP dependent negative supercoiling of the process. Next, in the case of, as you already know that DNA strands are antiparallel, 5 prime, 3 prime and 3 prime, 5 prime, and always run in opposite direction. DNA replication is found only on 5 prime, 3 prime directions. And in eukaryotic cell, 5 prime, 3 prime parent strand, on this strand continuous DNA replication is found, and only one RNA polymerase is generated, whereas on 3 prime, 5 prime parent strands, DNA replication is discontinuous and small fragments are produced in 5 prime, 3 prime directions. These small fragments are called Okazaki fragments. And DNA gyrase, after completing the DNA replication, DNA gyrase seal these small breaks between the Okazaki fragments. Whereas in the case of prokaryotes, RNA primer is removed by DNA polymerase 1, and in eukaryotic cell, RNA primer is removed by DNA polymerase beta. And this is the picture. Now you can see the picture. These are parental DNA template, 5 prime, 3 prime and 3 prime, 5 prime. Both are anti-parallel, and this is topoisomerase which unwind these two parental DNA templates, and helicase breaks the H2 bonds which are found between the nitrogenous bases. So these two strands get separated and form white shape forked. And in the 3 prime, 5 prime, small fragments are produced. There is discontinuous DNA replication is found, and very small fragments are produced. These small fragments are called Okazaki fragments. They are produced in 5 prime, 3 prime directions. After some time, DNA ligase seal the Okazaki fragments, and this strand is called lagging strand. And on the other side, 5 prime, 3 prime parental DNA template, only one RNA primer is required, because DNA replication is continuous from this side to this side in 5 prime, 3 prime direction. This is called leading strand, and this is DNA polymerase. Whereas and in the lagging strand, or in the 3 prime, 5 prime side, discontinuous DNA replication is found, and every time when a small strand is produced, every time RNA primer is generated. So this type of DNA replication is found in eukaryote, and In prokaryotes, RNA primer is removed by DNA polymerase. So after completing the DNA replication, RNA primer is removed. In prokaryotes, it is removed by polymerase 1, and in prokaryotes, it is removed by DNA polymerase 1, and in the case of eukaryotes, it is removed by DNA polymerase beta. And the size of the Okazaki fragments in prokaryotes 1000 to 2000 nucleotides long, but in the case of eukaryotes they are small in size, they are short up to 100 to 200 nucleotides long. So in this picture you can see DNA replication in prokaryotic cell. This is circular DNA, and only one single origin of replication is found. This is replication bubble. One replication bubble is produced only, and the replication takes place towards both the side. This is parent template strand, and this is daughter strands, and replication is taking place towards both the side. After some time, two daughter DNA molecules are produced. So these are the main differences between the eukaryotic and prokaryotic DNA replication. Thank you for listening me. If you like my efforts, do subscribe my channel. Have a nice day.